engineered mouse models of melanoma, but then now have moved more towards models where you can evaluate anti-cancer immune responses. And I think that this is something for a lot of folks that you're probably seeing some part of, and hopefully this will be useful from that point of view. And in the two different hats that I wear in terms of being a pathologist, sometimes for other people, but mostly for my own group, and then designing and building models, as pathologists, it's all too frequent that we get consulted at the end of a study, a box of slides shows up on your desk and you're asked to, do something that's very constrained in terms of, what they want to see. And I've always advocated that as pathologists, we need to really inform the people building the models as well for what would make, us do our jobs better and how we would do that. And so just to give a little background on some of the modeling parts, as you're all very aware that syngeneic or mouse models and xenograft mouse models are probably the workhorse for anti-cancer drugs in the pharma industry, but also in academics. And it's usually really mainly because they're cheap, easy, and fast to use. And pretty much most groups can do this. And they typically involve subcutaneous injection of cells. But there's a lot of problems with them potentially, but they've also been pretty useful in terms of moving the field forward in ways that have been sometimes confirmable, but they've been limited to relatively few lines that weren't really clear how many of them arose in the past. So one of the things that I'd hope as pathologists you'd also push back on is, and as reviewers of studies like this, is, how do you actually tell that something happened in these models? Sandy was talking about how to look at the tumor microenvironment. And those were a lot of things that are correlative. But in the modeling part, you have the ability to do functional studies. And I think, that part is something that you really should be doing better than you might be able to do in humans. So typically what we see, especially in the B16 kind of papers, is you'll see tumor volume plots where you have, let's say, 10 mice that were not treated or treated with a control, and 10 mice that were treated with an experimental drug, and you look at the tumor volume as a function of time, and you create error bars for each point in time, and then you see if there's a significant difference between the two curves based on the error bars. But they're usually like 20 days to 25 days is the end of the experiment, and almost all the time, the experimental curve still is growing, and that would be progressive disease in humans. That would be a failed clinical trial. It's just growing slower, but it's still growing out. And what the best endpoint in human clinical trials is that you extend survival, either progression-free or tumor-specific survival or overall survival. And this is possible to do in models, and this is what we try to strive for in the various things that we do. We try to tweak the model so that that's what we're looking for. And the main parts of the pathology that I'll present in a couple seconds are what that response looks like in a fairly, time point intensive fashion in terms of looking at lots of things. Just by euthanizing and looking at H&Es, you can see quite a bit over that time scale and sort of give you a little bit of a peek if you haven't seen it at that different time scale as to what one might expect. And a lot of correlative studies get done, for instance, like quantitating T cell infiltrates by flow cytometry. And a lot of times people are obsessed with getting enough events and cells for flow. So the tumor is typically ulcerated and larger than a centimeter, even if a centimeter was their legal volume for getting it, because they can extract a lot of stuff. But that probably is a really bad time point to look at and doesn't really tell you a lot. So here's just sort of an example of what I was just talking about with tumor volume plots, where you might have a control curve here in blue, experimental curve in green, or you could have an alternative experimental curve here in red, where the green and red end up at the same volume at a certain time point, but there's a different kinetic to it, where for a long period of time, the red curve seems pretty well controlled. And if you took histology and sampled stuff for experimentally at the flat part of the red curve versus the end of the experiment, it's almost undoubtedly that you see different results histologically as well as flow or other correlates. And that rarely gets done when tumors are smaller. And the great part about histology is you could look at a couple cells. It could be a very small tumor to look at, and people are typically unwilling to do that earlier time point evaluation. So, what we strive for, and some others as well, would be that you have what's called an established tumor. And this is a bit of a joke, I think, in the field. Usually this is about 7 days, and the tumors might be 1 to 2 millimeters. I don't know how that really is established compared to a human tumor, but if you start therapy after like day 12 or so with checkpoint blockers and almost all of the syngeneic models, the responses drop off to almost nothing. And it's not really understood why that is, but that's clearly what happens in most people's hands. So in the best scenarios, typically you'll start therapy at around day 7. The tumor will continue to grow for a couple more days to day 10 or day 12. And then by 2 to 3 weeks, the tumor will be gone. And histologically, when you look at the site, there won't be any tumor left. The mouse will live a normal lifespan if you let it go that long. And if you challenge with the same tumor, even at much higher doses, you can see a very exuberant memory response typically in these contexts. So those are some of the aspects that one would be looking for. I'll go through the next couple slides pretty quickly. We've made a number of models that are used pretty widely that are genetically defined, which can be useful in certain situations. The histology of the different models is kind of cool because you can actually tell the genotype to some extent from different models from other ones. For instance, the BRAF P53 melanomas are much more pleomorphic, and they're very, very clearly different from the BRAF P10 melanomas in a blinded fashion. However, other versions of this, it's not that clear. Sometimes BRAF CDKN2A versus BRAF P10 is a little bit harder to tell, as an example. I won't really show that per se. So one of the things that we found was that these lines, when we injected them into wild-type black 6 mice after they were grown and back-crossed to be syngeneic with B6, is that they typically will grow out and form tumors. And if you treat with checkpoint blockers, they won't actually have a response that you can measure. Although there's an exuberant T-cell infiltrate in the tumor, it isn't effective. So what we ended up doing to help with that is we mutagenized the lines by UV radiation and then single-cell clonal selection. And when we do that, so Yale University mouse melanoma is the YUM, and then YUMmer, it was a student named it, was exposed to radiation. So in wild-type BLAK6 is the unmutagenized version grows out. And I'll talk about the histologies of these in just a second. While in YUMmer 1.7, the mutagenized clone, you tend to see that it gets rejected at low cell number injections pretty reproducibly. And RAGs, they both grow out. You also can do T-cell depletions, which is another way of showing that there's a T-cell dependent process. And it's a little harder to see, and the control would have gone away, but both the CD4 and CD8 will allow escape in that kind of context, which is, I think, an important part. And I don't know if other folks are as aware of this. I think it had been known and modeled pretty extensively in the past this way, but for some reason in mouse models, the number of cells that you inject really affects whether or not tumors are spontaneously rejected. And so in our hands, for this particular model, at injections at around 100,000 or less, we can see spontaneous rejection fairly reproducibly, but then at higher cell numbers, we get outgrowth every single time. And that is typically our model, the outgrowth model for what we will use to treat. So we know we're pretty close to that tipping point for whether something can be rejected, and we kind of go for that. And so here's an example of a Kaplan-Meier plot where we go out for quite a long follow-up in terms of time. And you can see when you're obviously at the top of the line, you get 100% survival. That means the tumor's been rejected and you have the mouse living. And if it's euthanized because of tumor volume, that's the isotype control here, and we don't really see that. So you can get curves like this, and we use spider plots in addition to the Kaplan-Meier plots frequently to plot out the data rather than tumor volume plots when possible. So to get to histology now, so this is what we ended up doing in evaluating these models was doing timed euthanasias like at 4 hours, 12 hours after the cell injection, but then 1, 2, and then every day in the time course up to about day 24 or so. And there were neutrophils and macrophages within the first 6 hours within the tumor. We didn't see T cells or B cells until after day 7, at least at any real volume number. And I didn't really see what I thought was immune-mediated killing, either by histology or by tunnel or cleaved caspase 3 staining before day 8, and really not extensively before like day 11 or 12 with PD-1 or CTLA-4 blockade. But different therapies have different kinetics, and that's important. And then I could histologically tell whether a tumor looked like it was going to be killed entirely or would escape by about day 15, which is later than I thought it would have been in a blinded fashion. And the escaping tumors, you still see a lot of T cells. People talk about hot and cold tumors that are still pretty hot from that point of view. It's just that they're not effectively being rejected. And I'll show the images, and hopefully people will comment, and I'll solicit a little bit more in a second about what we're seeing in the histology. But flow cytometry really doesn't work that well for capturing this because the tumors are smaller than typical flow size tumors, and then also you get edge with middle, and there's a huge difference between those two things, which we can talk a little bit more about. This is one of the tumors that it doesn't matter at this time point whether it's going to be rejected or not, but these are basically all fairly uniform melanoma cells. There actually are a fair number of histiocytes or macrophages in the tumor in this view. I won't show the histology, but there's also quite a few at the edge of the tumor, which is over on this side. This is a tumor that will be rejected in about another 5 days. And you can see there isn't really that much death that's going on here. You can see in a couple other images in a bit. We see some neutrophils that are also coming along the edge of the tumor here. There are interspersed lymphocytes, which are by staining in different sections, are T cells predominantly. But the tumor cells, which you can tell by the chromatin and the nuclear size, still look generally reasonably happy at this point in time.
So Marcus, remind me, what is the origin of this Yummer cell?
So this is it's a BRAF activated, P10 deleted, and CDKN2A deleted melanoma model, which then had UV mutagenesis, so it was just irradiated with UVB in culture, and then single cell clones were picked out of the mix that were UV mutagenized and isolated as such.
And this is one of those. Black 6 background at this point?
Yes. Well, and so when you do the exome capture and sequencing, there are probably about almost 300 SNVs in the parental non-mutagenized line compared to black 6. So there are already things that are not, because when you have 4 alleles, the tyrosinase CRE, the BRAF, P10, and CDKN2A, there's chromosomal segments adjacent to those alleles that don't get crossed off fully. So you get things, and this is not uncommon, even with 10 generations or more of back crossing, there are still differences genetically between that and black 6.
Right.
This has about another 1,200 non-synonymous mutations, and maybe half of those get expressed probably by RNA-seq. So it has more from that point of view. But thanks. And so 5 days later, the same tumor, you can see that it looks a little bit more heterogeneous here. And I'll show a stain for these cells are labeled with GFP as well. But the tumor cells, you can still generally see as the larger cells here. Most of the other cells around the side are macrophages. That's probably a tumor cell that's got sort of pycnotic nuclei and has cytoplasmic contraction that's happened. But the morphological features of death here aren't really that classically like what you might see in apoptotic cells. And pathologists are actually pretty good at looking at morphological features of death. And here's to say an older dead cell probably here is a pycnotic nucleus. And you can count those and they get resolved. I'm not sure it's the best metric of stuff, but certainly they're there. But we haven't really been, we'll talk a little bit more about the mechanism of killing and how to morphologically look at that. But this is one version. So this is a GFP immunohistochemical stain, which in our hands works really, really well. It looks messier here because there's a lot of cleaning up, but we also have nuclear labeled tumor cells that are not being killed, and that staining is very clean. So this is probably cellular debris that's been phagocytosed by macrophages, and then here's the remaining viable tumor cells at the end point of when there's still some tumor left, and that will be resolved in a couple days.
You've got some large cells near the bottom that I would have guessed were tumor cells, but
yeah. So like these guys down here, exactly. So it's harder to know. I think the chromatin, which is a little washed out here in the hematoxylin stain, but if you look here, it's a little bit like, for instance, some of that I wouldn't know for sure what that is, or perhaps those do look like tumor cells to me, though, I'd be honest as well. But I think this is where the stain sometimes can be helpful. And in contrast, the last two images were things that could have either been treated or just spontaneously were regressing. When a tumor grows out, in the original version of this line, we saw, a big increase in mitotic activity, which you can see here. This looks like a very healthily growing tumor with, it looks like it's mostly tumor cells. Maybe a little bit of death here. It's hard to know if that's a vascular death. There's a couple spots where I can definitely see pycnotic cells. Whether they're immune mediated or not, it's harder to tell. But then what was surprising to me is this is an F480 stain of that same section. And it seems like there's 200% of cells. Like you have all of your tumor cells there, but that there are processes and even nuclei of macrophages that are absolutely everywhere within that section. And I remember seeing that in human tumors too, and really sort of being blown away by the density, which your eye just really doesn't pick up the same way. I'm sure, there are ones that you can kind of see that probably are as you're looking around, different spots here, but there really are much more, less catchy to your natural, at least for my natural ocular focus. We do also CD3 stains. There's probably about 2 times as many cells as you see here for CD3 staining of a tumor that's growing out in this model. We also see a big increase in the FOXP3 to CD8 ratio when things grow out. In our hands, in the mice, the FOXP3 stain works quite well. And we also use models where you can deplete FOXP3 with diphtheria toxin receptor models. And those tumors can be regressed at a fairly large size. So if one were to guess, or I were to hazard a guess as to what's important for the, blocking regression, I think the Tregs really do seem to be playing that role. And I just thought I'd show this in contrast to what we were seeing before, where at Yale we do a lot of preclinical work on this drug as well, but also have a clinical trial that's happening in humans. And so this is an agonist CD40 antibody. This, I think, is just given alone or with PD-1 blockade. And this therapy was started only 1 day before this image. And we see a corresponding systemic spike in cytokines, that go through the roof. Like CXCL10 will be up 1,000-fold in 1 day with this therapy. And it's a really wild phenotype from a histology point of view, because there's congestion and thrombi within the tumor vessels. There is, and this would be helpful too, we can maybe talk about this a little bit later, but in these kind of images, I have a little bit harder time telling neutrophils from karyorectic debris of tumor cells. In humans, I can generally do that pretty well. And in some of these images, I find that I think it's karyorectic debris, but it's a little bit harder to absolutely be sure just with the H&E, at least with our staining. It wasn't really appreciated. And the other place we see what might be a vascular phenotype is actually in liver. And I was wondering if anyone has seen anything like this before. I only have one image here, but I can kind of describe the rest of it. But what we see within 2 or 3 days what end up being infarcts more peripherally in the liver. This could be one of those with histiocytic infiltrate after the infarct has happened. You can see what's probably a central vein that has, there are ones that have unequivocal thrombus, and here it's a little bit harder to tell. but you can also see there's a lot of macrophages by staining that are lining up on those vessels, congestion elsewhere. And I don't know, anyone want to make a guess at what these cells are? I don't know, people that, you know, they're smaller. I thought, well, they could be histiocytes. Could they be NK cells? What?
Could they be EMH?
I don't think so, but that's a fair. So this is, yeah, these will be like 6 to 8 week old mice, but I suppose that could be. This is 1 day after the drug is given too.
Oh.
So, and I'm thinking that it's probably infiltrate rather than proliferative. But
I mean, I would
so
guess T cells, of course, but
right.
I could be wrong.
So that's what I was thinking. So they actually end up being B220 positive. I think that might be the next slide.
Mm-hmm.
So here's like the granulomatous histiocyte things. I just wasn't expecting that because they were negative for NK markers, for T cells. And I was like, what else could they be? So I guess the question I have to the group is, B220 is often used as a B cell marker, but is there other cell types? Would a dendritic cell subset, you know, what are people's thoughts, you know, for that kind of group being positive as far as being B cells or not?
Are there some eosinophils mixed in there too?
I think there are some neutrophils. I don't know how many EOs there are, but that would be, yeah, from an allergy point of view, potentially. But I have to admit that I don't know that I am the best person at recognizing. As a dermatopathologist, I'm looking for them every day on the human clinical side, but on the mouse side, I don't know that I've had that many positive controls that I go, yeah, that's definitely an eosinophil. As opposed
we
to
had a really nice session, which I think you probably, and this dates back a year or two on mouse eosinophils, and the gist of it is there's a bunch of different types. Remind me, who gave that? It wasn't you, Jerry. It was maybe it was the other Jerry. He's keeping quiet. Anyway, mice have more complicated eosinophils.
Right, and
I wish I recorded that session for you.
We can do, like, so the question for a lot of this stuff too is we do a lot of single cell RNA-seq, and we can do a lot of fingerprinting from that. Not that you'd have to do that for something like this, but sometimes there's cell types or cell states that may not exist exactly elsewhere, and it's not a bad way to do that. But this isn't interesting enough from our point of view. It's just kind of a cool thing. Humans do have liver function test abnormalities, but no one ever does the biopsies to see if they have this particular issue in terms of liver endothelial or thrombotic part. So CD40 is the target of the antibody. It's expressed on macrophages, dendritic cells. It has a B cell phenotype in terms of lack of class switching if you lack it, but it's also expressed on endothelial cells. And so it seems like that certainly is one of the things that one could be seeing, but wasn't really expected at all here. And one of the cool things about this is that even though you're seeing all the B cells in here, we see a comparable phenotype, but lacking the B220 positive cells, which I guess means they probably are B cells in rag mice. But in rag mice, we actually see almost a more severe phenotype with this agonist CD40 antibody, where it can be dose limiting or lethal in some situations or rare situations, but high dose steroids entirely block that phenotype, which, you know, people usually think of those working on lymphocytes, but they completely reverse this innate immune phenotype. And that's the only thing that's semi-confidential, I guess, in the talk, but we're interested certainly in figuring out how that's happening. And just, I thought,
this is Jerry.
Hey, Jerry.
Just go back to that last one. I think I always thought those focal areas of the B220 were compatible with, in the other slides, you had foci of small lymphocytes. So it might not be the histiocytic lymphome. But go back a couple.
Yeah.
You'll see you have foci of lymphocytes in the dark. Yeah, I'm not trying on the left. Yeah, so those might be the B220 positive.
Yeah.
And they look almost any plasma cells in there?
Not that I could, no, I didn't really see that. It's also maybe a little bit early because this is like day 11 after the tumor was ever there. So in terms of, I don't know the time course for plasma cells as well, But we don't see any B cells typically in tumor lesions. This is liver, not tumor, before like a week. So in terms of expansion of antigen-specific B cells and plasma cells, it might be a little bit quick.
Okay, another question I have, you mentioned at the beginning. So in a lot of these tumor models, when you have an immune response or any inflammatory response, the most severe response is on the edge or the outer part of the tumor, not in the middle or inner side. But numerous publications, they're not pathologists, and they don't know that, so they measure overall. And so could you say something about that?
Yeah, so we see it in the middle. I'll go back a few slides to, this is basically the middle of the tumor now for the Yummer models where we use different therapies. It can be a little spotty across the tumor where there's still pockets of tumor left in certain spots, but then it's clearly regressed in other spots. But then it's really almost like that was on cue, Jerry, because the next slide I was going to show is this is a B16 tumor that doesn't have any model antigen expression. And this is a CD45 stain. And I have never in my life seen a tumor that has less white cells in the main part of the tumor than this. People use also different versions of B16 where it expresses ovalbumin or other antigens, and those do have some T cells in it. But basically, you have to search miles to pretty much get T cells on the inside of the tumor. There's a couple, if you look closely here, a couple of histiocytes that are kind of scattered throughout that are, few and far between. There's probably one over here. Oops, sorry about that. But what people do is they do flow cytometry with no histology, and this is what they're getting. They get the skin of the balloon, if you want to think of it that way, when they're taking stuff out. And so this is like an azure blue counter stain, which turns the melanin from dark brown to like a greenish color to bluish color. And this is the melanin here. And a lot of times people don't like using B16 because it's almost impossible to read IHC through it, even with the red reaction product. It's really hard to see. But
that's been my experience.
Yeah. So I mean, it's funny because people tend to try to do bleaching and things like that. But in my hands or our hands, this azure blue counterstain works way better for heavily pigmented tumors like B16. Which can be a little bit variable, but it's really striking because this is, the outside of the tumor edge, and what you were saying, Jerry. And sometimes B16 can respond, but I don't have it's always they don't give you enough time points to see what's going on in the middle of it. And I think that's kind of the key part is people want to have their cohorts to determine outcome for stuff. But if you're trying to figure out what's actually going on, you really need to have people go back and give you a couple more time points along the middle of the time course. It doesn't have to be tons of mice, but just a couple mice perhaps each. And I think that, let me see. Oh yeah, we also do some left ventricle injection modeling where you would just inject by visual cues into the left ventricle. And then you get this is 5 minutes after injection, you get luciferase positive signal all throughout the mouse that wasn't there 5 minutes before the injection. And then like a week later, you start to get concentration in certain spots and loss of signal in most of the other spots. You can cycle some lines and get relatively brain-specific variants of it, which we haven't pursued as much. But what's kind of cool with the brain versions of it is that unlike direct injection, where you're doing a burr hole and using Hamilton syringe to inject 10 microliters of tumor, something like that, into one side, you get multiple lesions here. And we've been trying to do the histology of this kind of stuff with small lesions. It gets quite challenging if your density is not enough to actually, you just have to do lots of sections to actually hit something that you really know. These lesions are a bit bigger. These would not be hard to find. But for the small ones, we have relied more on stereo microscopy with fluorescent tags, which you really wanted to, but then also trying to go back and stain for those things. And also look at immune responses and these kind of things, because that's a little bit more relevant from compared to humans in terms of, human melanoma. You can almost always excise the primary lesion in the skin, but it's the metastases that are the problem clinically. I think mostly that's what I have in terms of the slides. It was mostly just that in case some of you are doing other people's pathology work, just to try to push them to give you all the things you need to have to be able to evaluate what you're looking at. And you know, one of the things we tried, which I thought was two things we tried, which was surprising to me on a functional side, is we grafted into B-cell deficient mice and saw no difference in phenotype. It got rejected the same frequency. And there's a few Nature papers earlier this year where they suggested that B cells correlate with T cells in human things, but it didn't really evaluate them functionally. And then the other part, which was surprising, is that we grafted into perforin deficient mice. And we thought that was going to be a control because if you ask immunologists, most of them would argue that perforin, which is a pore-forming protein secreted by NK cells and sometimes by CD8 cells, is secreted in a TCR-dependent way when T cells, CD8 cells meet tumor cells, and that, you know, punches holes in the tumor cell, and the granzymes come in and cut things up. So we expect to see no tumor rejection. And it ended up happening that we actually saw a really markedly enhanced tumor rejection. So the exact opposite phenotype. And we thought we genotyped wrong. We went back and looked, and what we ended up finding was that there's a literature on perforin deficiency, which also suggests it's variable depending on the model. Class 1 deficient models that are NK dependent typically don't do well when they're in perforin deficient mice. So that's probably true for NK cells. But there are humans who have perforin deficiency who have hematophagocytic lymphohistiocytosis and like an activated macrophage syndrome. And that seems to be what we're seeing in these mice in terms of seeing the enhanced rejection responses. So it kind of leaves me, and I think I've asked around a ton for that we really don't even know how T-cell mediated killing is happening mechanistically. And so from a pathology point of view, this is an opportunity also on the morphology to see what, you know, that's why I was looking at those dense time points to see what you can get as far as ideas go. But we're trying to do a little bit more on that space, and I have a couple of ideas, but it's still kind of a wide open area.
Do you have pictures of that
porphyrin
porphyrin deficient situation with macrophages engulfing tumor cells? That would be.
Well, so I don't. They look kind of like the image that I showed at day 15, where there's just a few tumor cells. So it's not like amperipolesis, where you're getting everything getting eaten, right? You know, which we see like for Roside-Dorfman or something like that we can see that phenotype in. But we did single cell on the perforin deficient mice, and it looks, when we have effective immune responses, they all kind of sit on top of each other, including the macrophage part. It doesn't really separate out. So I think we're seeing an activated macrophage syndrome like phenotype in anything like the immune checkpoint, things like that, in that it's probably interferon gamma dependent, and that's the kind of things we're seeing. But histologically, it wasn't that different from what I was showing in terms of the time points. Jerry, you had to...
I had a question. Did you do any, with your mouse models of melanoma, any anti-angiogenesis therapies? Yeah,
so I have in the past, not with the immune therapy side of things. One really cool experiment that we had done once was we had, with Linda Chin, expressed VEGF, the opposite really, in one of these models, sort of like a cell line model. And we had a massive like liver peleosis phenotype where the vascular channels in the liver were just massively expanded. And what seemed to have happened was a systemic increase in VEGF just made whatever vessels could respond to that really take off and grow.
So it didn't matter where you were expressing it from, it acted systemically.
It set that off. We tried some stuff back then for it didn't work. People have tried straight up VEGF receptor inhibitors and antibodies, which haven't been effective. But right now there's several kind of dirty tyrosine kinase receptor inhibitors that are seeming to have efficacy when you combine them with PD-1 blockade. People have called them VEGF receptor inhibitors, but they're not. They inhibit FLT3, they inhibit all sorts of things you probably wouldn't want to inhibit. But for some reason, some drugs just seem to have the right combination of things they're hitting, and people don't really know what the target is. Why do you ask that, Jerry?
Well, you mentioned earlier the CD40, you had thrombosis and necrosis associated in the liver, at least. So, I mean, you could have multiple mechanisms. Antiangiogenesis could also be targeting endothelium, even when the vessels have developed already. I had one experience with a targeted therapy to the blood vessels in the tumor, but it turned out to be blood vessels outside of the tumor causing thrombosis, and then the tumor became necrotic and shrunk. But the people that I work with didn't understand their whole concept, and it's in clinical trials now, but it has potential for thrombosis.
What was striking here was the agonist CD40 antibodies seem to only be hitting the endothelium in liver and in tumor out of all the places we looked, throughout the body. And unlike the liver, which had geometric infarct-like areas, the tumor didn't really have geographic necrosis like you might expect to see in an infarct. It was much more piecemeal here and there, and it was really exuberant over that short period of time. So we're still looking at mechanism of action, but right now it seems like it's cytokines. The cytokines go through the roof with this drug in humans and in mice, and we see the same kind of cytokines go up in both. But it seems like we're kind of also revving up macrophages to help and it's also T cell dependent, the rejection response. So if we deplete T cells, we don't see that rejection anymore. Just to go on briefly, I do a lot of human work too, and the problem with the human work is that we never get that day 15 biopsy that I was just showing for the mouse, practically ever. What we get as part of standard of care is like a patient's treated with a checkpoint inhibitor, 5 out of their 6 masses go away, and the 6th one doesn't go away, and you get that taken off because it's medically indicated to clear to the patient of disease. So that's the one failed response in the context of something that was an effective response. And what we're trying to get set up are these window of opportunity trials or neoadjuvant trials where you do a biopsy before you start treatment, and then you do a biopsy or resection like 2 to 3 at a time point when the patient's on therapy, and then you follow another mass in that patient to see if he actually responded or she at a distant site. And those would give us a little bit more understanding about what's going on in the actual mass in the human responses, because the amount of those specimens that you can get your hands on is really, really low to absent in almost every place, because you're putting the patient at risk for a procedure they might not need, and there's no money to pay for it unless it's a clinical trial. So those things usually be the things that block people from doing it.
Okay, does anybody have stuff they want to share today? I've got one thing I can show you, but the floor is open, so I'm happy to entertain whoever's got some slides to show. Raul?
Yeah, I just had a question still for in line of this presentation. We are getting more and more involved in doing multiplex stainings that should sort of mimic flow analysis, and I'm sure that you're also getting this kind of request. And one of the primary cell types that is eluding me at the moment are the Tregs, the regulatory T cells, and the NK cells, which I understand to be more of a transient phenotype of T cells respectively, so macrophage-like cells. And I'm wondering if you could comment on that, because on flow, this is a little bit different with rather multiple channel flow. How would you address that, if at all, in an immunohistochemical approach?
It matters whether you're talking about mouse versus human. So
mouse.
Yeah. So with the mouse stuff, NK 1.1 works like every other time for us. It's not great, and the staining isn't very pretty, but we can see, we know we have NK cells because we do single cell RNA-seq, which is much more, or the flow-based approaches. There's actually, if you look, we just had a paper that came out in Nature yesterday with a collaborator, but it's Ring is the senior author. I'm on it, Bosenberg, but it's on IL-18, and in that paper, we actually show that a version of this drug works on class 1 deficient tumors, and it's totally NK dependent. So it has a ton of NK flow. I don't know that it has the IHC as much, but it's not a great marker, but it's what we have. in humans, CD56 works better. It's not only specific for NK cells, but NK 1.1 is what we use, and it's about the best that I have. For macrophages, F480 typically seems to work, although there's some subsets that are F480 low where you're not going to see them as well. And the Tregs, I can send you. You can follow up. Whatever antibody we use for Treg staining works really well on formalin-fixed paraffin-embedded sections typically.
Okay, thank you.
What was the model in that?
Looking forward to your email.
What was the model in the IL-18 paper?
So it was a Yummer model I just showed you, Yummer 1.7.
Cool.
And then we also did MC38, CT26. I mean, it's nature, so you have to do like 8 models, you know, and it's like, you know, I think it's 29 pages with the supplemental data. There's a lot of stuff that went into it, but I think there's at least 2 or 3 class 1 deficient, like beta 2 microglobulin deficient models that we made for that.
Super cool. Okay, I'm just going to present briefly something that's come across my desk recently, which brings up sort of a new aspect of tumor immunology that I think is worth a brief discussion. So just briefly, the root of this project is this tubicin mouse organoid transplantation system. As you guys all know, I'm a big advocate for syngeneic transplants as a way to be studying a more complex biology that includes the immune system, but do so more efficiently. So my labs, as you know, generated several syngeneic transplant models, both cell lines and precancers. So I was happy to see when this paper came out, and they did a similar thing with pancreas. They go through the details, and I won't go through all the details with you, but basically this is a genetically engineered mouse, tubicin engineered mouse originally, that they have both KRAS and P53 knockout engineered into the prostate ducts using the pancreas Cree model. And then the collaborator that I'm working with now has added yet another layer on top of that, which I'm not going to talk about because that's his proprietary secret. But I will show you, well, sort of interesting. This paper shows these different stages with different degrees of progressive epithelial changes, from early lesions that are maybe normal or Pannin-like through cancers. PDA stands for pancreatic ductal adenocarcinoma. So they argue this looks like pancreas cancer, and I would say they're right, it does. And then metastases, peritoneal and diaphragmatic metastases. This paper goes through a few markers that are interesting, including the vascular markers, but they don't talk about the inflammatory pattern, and they don't talk too much about the contribution or strain background. So just like Marcus talked about, this is all back-crossed onto a BLAC6 background, but the original ES cells were not BLAC6. So in order to continue to have your transgenes or targeted genes, you have to carry with it a little bit of that original 129 DNA adjacent to those loci. And then when you do these tumor-derived transplants, they're not going to be completely syngeneic. There's going to be a little bit of residual ES cell strain. The other thing they've done is they've incorporated a GFP marker, and I think that is something we've talked about previously. GFP all by itself can have antigenic properties. In any event, the reason that I wanted to bring this up today, these are just 2 examples from the cohort that I'm looking at right now, and you can see it does have that phenotype, which looks like a pretty good mimic for pancreatic ductal adenocarcinoma. But what I was struck by is the degree of neutrophil infiltrate here, and I'm trying to zoom up so it's easier to see those neutrophils, but I think you can see segmented small nuclei in here. And I think there's a significant component of the inflammatory reaction that is neutrophils. This is not something I'm used to seeing in humans, although people do talk about it, but I think they talk about it in part because of the animal models. And I'm curious to know people's thoughts about neutrophil infiltrates and how that pertains to what we're trying to do to study the tumor immune microenvironment. Open question.
Hello, Jerry again. There are some mouse studies. The mouse studies that show depletion of neutrophils in a mouse model of a transplant or a carcinogenesis study results in modification of the growth of the tumor. I can get you those if you want.
Yeah, it'd be good.
I've seen people report both ways, and I've actually had trouble fully depleting neutrophils from the tumor itself. So I can deplete them in peripheral blood and things like that, but I, like macrophages, I haven't really been successful in getting rid of them intratumoral neutrophils in total. I agree with you, Sandy. I do see neutrophils in my hands. It's in the context of effective responses that I see them more often, or inflamed environments that are, where I think there is some killing going on, but maybe not enough. In human tumors, I'd say like in some squamous cell carcinomas in skin and other places, you can see them occasionally where there'll be lots of neutrophils around, but most of the time it's not a prominent component in human tumors.
Certainly most of the mammary syngeneic transplant models that I work with, we see when we get a good immune response, which we have been trying to induce in various ways, we see predominantly a CD8 infiltration, lots of macrophages. We don't typically see neutrophils in that setting. I think when we do see neutrophils, we're always suspicious that we introduced something at the time of transplant surgery, like a few bacteria crept in there, or our sterile technique was not perfect. So that tends to be my suspicion. In this case, my suspicion is that maybe I'm getting a neutrophilic response to the GFP in particular. And I'm curious to know if people think that that's a thing. You'd expect GFP to act like a tumor antigen, if you will, sort of an induced antigen that's new to the host. And so you might see more of a typical tumor response with CD8 positive cells and whatnot. But I don't know the answer, which is why I'm bringing this up. And the other possibility is this is poor sterile technique in this model system, with whoever did these surgeries, introduced something they didn't mean to. But I think it's probably a real reaction. I see it in too many of the animals. And I'm just curious about understanding this aspect of immune reaction, a neutrophilic immune reaction to tumors.
Did you check if they are EOS versus neutrophils?
It's a good question. How would you recommend I do that?
Maybe there are markers you want to check. Probably I will try to send you that, because sometimes this immunocompromised mice, they may have that kind of response, having eosinophils.
Well, I should say these are intact B6 mice.
Oh, okay. Then
it's
I will withdraw.
Pretty much a syngeneic transplant, as they try to backcross their genetically engineered mice fully onto Black 6, but as we alluded to, they're never going to be complete.
Okay.
But I think it's still a good question with what we know about mouse eosinophils and the complexity of that.
How long after transplant was this?
Oh, you would ask me that. I have that written somewhere. I think it's about 4 or 5 weeks.
Okay, that's long. Yeah, that seems like a real ongoing reaction. In some cases, I've been wondering when you do find more neutrophils, not necessarily related to the technique and the sterility, but you always have to ask yourself the question of how much of these cells or tissue parts that you implant do actually survive. So they can be sterile cell death, but then I would expect more in the beginning, and usually this gives some scarring. So what you're showing now doesn't seem to be related to a lot of dead material being implanted.
So I will say, and I won't tell you what the experiment is exactly, but this is one of the experimental animals, and I think that compared to the control animals, we are seeing quite a bit more heterogeneity in the epithelium, and this is the interesting thing that we're trying to pursue There's some question about whether we're seeing more of a squamous metaplasia, and trying to separate out whether that squamous metaplasia or squamous-like metaplasia is associated with greater inflammation or is just part of the heterogeneity of the model. And I actually favor the latter just because the inflammation seems to be pretty consistent in these experimental animals, and the other one I showed you was a control. You know, this turns out to be important because we're learning that pancreatic cancers in humans, when you try and subtype them, the two main subtypes are sort of a pure ductal subtype and a subtype which begins to express squamous keratins. And whether you want to call that the beginnings of EMTness or whether you want to call that a true squamous metaplasia, it's not something that we as pathologists would call squamous carcinoma by any stretch, but I think that it becomes very interesting to think about. So I'm also curious about your thoughts about whether you would refer to some of this as sort of early, I don't know, metaplastic change in the tumor and even squamous metaplastic change in the tumor.
I think in some of the larger duct, I mean, it's when you get multi-layering, you know, and, you know, if you have some of that in some places, that could be the beginning of that. It's kind of like, you know, where you have like GE junction changes, not that you're going to have, you know, mucinous or other changes that correspond to it. But one of the things I was going to mention too, Sandy, you talk about GFP and the relative antigenicity of GFP. So we've expressed a lot of things in our models, and we don't actually typically see GFP making something get rejected when it's not normally rejected otherwise. And we've done a fair amount with that. In contrast, the puromycin acetyltransferase gene, so the, you know, puromycin resistance, we can't get that to grow in some contexts at all. So we actually had to go in doing a CRISPR screen and had to basically pay a couple grand to get puromycin tolerant mice bred. So that, and that worked. So it really was, we tested it. It was that gene product that was causing the rejection. So some antigens are incredibly strong and will result in rejection every time, but luciferase and GFP and generally RFP probably only tweak it a little bit. If you have other things on board in combination, they might be too much. And even like model antigens like LCMV and ovalbumin by themselves may not be enough to induce rejection.
That's interesting.
But when we put LCMV antigens on Yummer, it gets rejected, but not on Yum. So it depends on the context as to what one's looking at. I'm always struck too by how cool the pathology is. This is like organoids. This is so neat that you get this diversity and this stromal expansion, you know, which we don't see in a lot of the other mouse models. And the GEM model was also very attractive from that point of view, as well.
We should continue to collect our experience with additional genetic engineered cassettes that provide some, none, or a lot of immune reaction. So we have seen immune reaction to GFP that in certain contexts can be limiting, but I agree it's not enough for a full rejection. We saw that in some tail vein metastasis. Well, tail vein and also spontaneous metastasis experiments we were trying to do. I've heard tell, but don't have direct data to compare, that M-cherry is worse than GFP with respect to antigenicity. And this is the first I've heard that the puromycin resistance gene is very antigenic, but I think that's worth noting.
It could save you a lot of money if you're doing it. I mean, it costs us like about a half a year to correct that because it was a CRISPR library that was in that. We couldn't get around that easily. So that's what we had to kind of work with.
All right. Any last words from anyone? We're at the end of our hour. I'll reiterate that some of what I just showed is preliminary data. So let me know if you want examples of this for something, for a review or something. We can probably arrange it, but you have to ask me. And similarly, you know, Marcus, we're going to all read your Nature paper, all 29 pages of it, plus supplementary material, because we have nothing better to do in this era of COVID, right? So do send a link to that. And the other thing, if you can send me a link to the method for the B16 counterstain. That's something that I'll adopt in my lab right away.
Right. That might take me a little bit longer because see if I get the histology folks to send that, but I can say the name of it and probably some links, but I'll get the exact protocol they use within a couple of days for sure. Thanks.
That sounds great. All right. Thanks, everybody. We'll see you next month. Please don't be shy. Any topic is fair game. I love this tumor immunology. We can keep talking about that. We can go back to COVID with some new publications coming out if people want to talk about that. Open to anything. So let me know.
Well,
Cheers.
thanks a bunch.